Welcome back, learner. Today we're taking a deep dive into G-protein coupled receptors.
GPCRs.
GPCRs, yes.
And we're going to be focusing on this sort of intricate dance they do with a family of proteins called beta-arrestins, and how that leads to this interesting thing called bias signaling, which has some pretty cool implications for drugs and drug development.
Okay.
Well,
yeah. I'm intrigued already. Before we get into all of that,
Sure.
let's just take a step back and refresh our memories about GPCRs.
Yeah.
Like, why are these a big deal?
Oh, they're a huge deal.
Okay.
These receptors are like the gatekeepers of our cells.
Yeah.
They're involved in so many processes.
Yeah.
Like, I mean, everything from how we see and smell
Uh-huh.
to regulating our mood and our blood pressure.
Yeah, they're really involved in a lot.
Yeah, a ton.
So it's no wonder then that they're really a big target for drug development.
Oh, absolutely.
I remember reading somewhere that a huge chunk of approved drugs actually target GPCRs.
Yeah, that's right. Over a third of all currently approved drugs target GPCRs.
No.
And it's because they're just involved in so much.
Mhm.
For example, rhodopsin,
Oh, yeah.
the very first GPCR whose structure was determined it's essential for our vision.
Right.
So
So
we-
that's amazing. Like these little tiny receptors are part of how we actually perceive the world.
Yeah, it's really
So structurally, what makes these things so unique
Yeah,
to be able to do all of that?
So imagine a cell membrane as like a wall, right? And the GPCR is like a winding staircase that goes through that wall.
Okay.
It actually spans the membrane 7 times, forming these characteristic alpha helices.
7 helices. Wow.
Yeah.
That sounds pretty complex.
It is.
Is that what allows it to bind to so many different types of signaling molecules?
Exactly.
Okay.
So on the outside of the cell, the GPCR has this binding site.
Okay.
It's almost like a lock
Right.
that fits very specific signaling molecules like hormones or neurotransmitters.
Right.
And these molecules, they act like keys. When they bind, it triggers this whole cascade of events inside the cell.
So it's like sending a signal from the outside world into the cell.
Exactly.
Right.
And for a long time, scientists thought that GPCRs worked primarily by activating these other proteins inside the cell called G proteins.
G proteins.
Yeah.
So another player in this whole.
Yeah, another character in the story.
Okay.
Think of them as messengers relaying the signal from the GPCR to other parts of the cell.
Okay.
And this activation triggers the production of second messengers like cyclic AMP and IP3.
Right.
And these go on to affect various cellular functions.
Okay. So we've got the signal binding to the GPCR,
Right.
activating the G protein,
Yeah.
and then triggering this cascade of events inside the cell.
Exactly.
Where do these beta-arrestins come in?
Oh,
I remember you mentioned they were involved in this thing called biased signaling.
right.
What is that?
So initially, beta-arrestins were thought to just desensitize and internalize GPCRs.
Oh.
Imagine them as the cleanup crew, you know.
So turning off the signal.
Yeah, essentially turning off the signal and pulling that receptor back into the cell.
Oh, so it's a regulation mechanism
Exactly.
to make sure that things aren't signaling forever.
Exactly.
Yeah.
But as research progressed, scientists discovered that beta-arrestins were doing a lot more than just switching things off.
Really?
Yeah.
What else were they doing?
They can actually mediate their own unique signaling pathways.
What?
Yeah.
So they're like a cleanup crew that's also starting their own
I know.
chain of events.
It's kind of counterintuitive, and that's where this concept of bias signaling comes into play. So how are those beta-arrestins doing?
Okay, so I'm still a little fuzzy on how these beta-arrestins can both turn off a signal and start their own separate pathway.
Yeah, it's a great question.
It's like they're playing both sides.
Yeah, and it really gets to the heart of how complex these systems are.
Yeah.
The key is that different ligands can bind to the same GPCR,
Okay.
but each one can sort of induce a slightly different shape, or what we call a conformation.
So like a key fitting into a lock,
Yes,
but subtly changing that lock's position.
exactly.
Okay.
Okay, so now imagine that a G protein can only bind to the receptor when it's in one specific shape,
Mhm.
while beta-arrestins prefer another shape.
So
Depending on which ligand binds,
okay.
the receptor can shift to favor either the G protein pathway or the beta-arrestin pathway.
Okay.
And that's what we call bias signaling.
Okay. So it's not just about turning a signal on or off,
Right.
but about directing that signal down a specific path,
Exactly.
depending on which ligand binds to the GPCR.
You got it.
Okay, I think that's starting to make a little more sense now.
Yeah, and this is where it gets really interesting for drug development.
Okay.
Because if we can design drugs that act like specific keys, ligands that bias the signal toward a beneficial pathway,
Okay.
we could potentially get the desired effect.
So fewer side effects.
Exactly, fewer side effects.
Wow, that would be a game changer for so many medications.
Oh, yeah, absolutely.
Can you give us an example of how this might work?
Sure. Let's take a look at the angiotensin II type 1 receptor,
Okay.
or the AT1 receptor.
Okay.
This plays a crucial role in regulating blood pressure.
Right.
So when the natural ligand angiotensin II binds to this receptor,
Okay.
it activates the G protein pathway,
Uh-huh.
which leads to blood vessel constriction.
Right. I remember learning about that.
Yeah.
So how does bias signaling come into play here?
Well, researchers have actually discovered ligands that can bind to the AT1 receptor,
Okay.
but they preferentially activate the beta-arrestin pathway
So instead of
that G protein pathway.
U protein pathway that leads to the vasoconstriction.
Exactly.
Right.
And what's even more fascinating is that the beta-arrestin pathway in this case
Yeah.
seems to trigger beneficial effects like reducing inflammation and protecting against heart damage.
Wow. So instead of just blocking the receptor altogether,
Right.
which could have its own downside,
Exactly.
we can sort of nudge it towards a pathway that's actually beneficial.
It's exactly.
That's really cool.
It is. And it's not just the AT1 receptor.
Okay.
The beta-2 adrenergic receptor, which is involved in our fight-or-flight response, and the mu-opioid receptor, which plays a role in pain relief.
Right.
Both of those also exhibit biased signaling.
Really?
Yeah, and those are being actively investigated for developing more targeted and effective medications.
So it's like we've opened up this whole new dimension of drug discovery.
Yeah.
It's not just this one-size-fits-all approach anymore,
Right.
but we can really tailor our strategy to use these nuances of GPCR signaling.
Exactly. And, this new understanding wouldn't have been possible without all of that research into beta-arrestins.
It's amazing how something initially thought of as just a simple off switch turned out to be so complex.
I know, right?
So how do scientists study something as intricate as bias signaling?
Yeah, it's a good question, and
It sounds really challenging.
it is, but they use a variety of techniques to kind of tease apart these pathways. One approach involves using specific ligands that we know bias the signal in a particular direction.
So it's like finding the right key
Yeah.
to fit that lock,
Yeah, that's a good analogy.
to activate the pathway you want.
Exactly.
Oh.
Scientists can also introduce mutations into the GPCR gene,
Oh, okay.
subtly changing the lock to see how that affects signaling.
So are there ways to actually observe these interactions?
Yeah,
Like to see which protein the GPCR is choosing to partner with?
absolutely.
Wow.
Techniques like BRET, or bioluminescence resonance energy transfer,
Okay.
allow scientists to measure the proximity of two proteins in real time.
So it's kind of like attaching tiny beacons to each one,
Yes, exactly.
to the G protein and the beta-arrestin.
Mm-hmm.
And then if they get close enough,
They light up.
they light up.
It indicates an interaction.
Wow. So they can actually watch these molecular dances happening.
They can.
Wow, that's amazing.
It's really cool.
So we've covered a lot of ground.
We have.
It's a lot to take in.
It is.
My brain is starting to feel a little full.
Let's,
I think we should pause here.
yeah, let's pause here.
Let all this information sink in for a minute.
I think that's a good idea. So I hope that information had a chance to settle in.
Yeah, definitely feeling a little more settled now.
Good.
It's amazing how much depth there is to this whole idea of GPCR signaling.
It really is. And, we've only scratched the surface here,
Yeah.
but hopefully it gives you a sense of just how much there is to learn about these fascinating receptors.
Absolutely. And I think the biggest takeaway for me is that these beta-arrestins,
Yeah.
they're not just passive players anymore.
No, not at all.
They're actively shaping the signals being transmitted within our cells.
Right.
And that opens up so many possibilities for developing more targeted and effective therapies.
Exactly.
So it really feels like we're at this turning point
Yeah.
in our understanding of GPCR function.
I think that's a great way to put it.
And I think this whole idea of biased signaling, it's really changing the game in terms of drug discovery.
Yeah, absolutely.
It's not just about blocking or activating a receptor anymore,
Right.
but about fine-tuning those signals to achieve the desired outcome.
Exactly.
So maybe this is the key to developing safer and more effective treatments
Yeah,
for a whole range of diseases.
it really could be. And, the research is ongoing.
Yeah, it's pretty exciting to think about.
It is.
Well, this has been an incredible deep dive.
Yeah. It has.
I've learned so much.
Me too.
I always find it fascinating to explore these complex biological processes.
Yeah, me too. And, I think it's really important to remember that we're still learning so much.
Right.
Every day, there are new discoveries being made.
Yeah, and maybe this will inspire some of our listeners to dive even deeper into this world of GPCR research.
Absolutely.
So for all of you learners out there,
Yeah.
keep exploring.
Keep asking questions.
Stay curious.
Because the world of science is full of wonders waiting to be uncovered.
And that's a wrap for today's deep dive.
Thanks for joining us.
We'll see you next time.